It is a method of growing a plant cell, either it might be a cell, plant cell, or a tissue or an organ in an artificial nutrient medium. The cells of the explant multiply and give rise to undifferentiated mass of tissue called callus. So autoclaving will kill all the microorganisms and it is fully sterilized. What are transgenic plants? Genetically modified plants are called transgenic plants. Hello everyone, welcome back to the last session 6 of this very important and interesting chapter called Strategies for Enhancement in Food Production. What are we going to study in this last session of this very important chapter? We are going to study about plant tissue culture. What is plant tissue culture? It is a method of growing a plant cell or a tissue or an organ in an artificial nutrient medium under aseptic conditions provided with controlled conditions of light and temperature. So what is tissue culture? It is a method of growing a plant cell, either it might be a cell, plant cell, or a tissue or an organ in an artificial nutrient medium under aseptic conditions. What do you mean by aseptic conditions? Under sterile conditions provided with controlled conditions of light and temperature. Hope you have understood what is tissue culture. Now the science and technology has improved to such an extent that you can do you can manipulate the genes and come out with good varieties of plants or good varieties of crops also, and all that is possible through a very important branch of science called biotechnology, and that too with plant tissue culture. So as I said, very important word you should understand. So what is plant tissue culture? It is the isolation or separation of cells or tissue from plants and growing them in a synthetic medium under aseptic conditions or sterile conditions. Very important. If the nutrient medium is infected, then it is not possible to produce the plants. So the person who discovered plant tissue culture was Haberland. Who is the person behind this plant tissue culture? It is Haberland. So what is plant tissue culture? It is a method of growing a plant cell or a tissue or an organ in an artificial nutrient medium. I'll come back to it. What is artificial nutrient medium? Under aseptic or sterile conditions provided with what? Controlled conditions of light and temperature. Moving on to a very important word called totipotency. What is totipotency? It is the ability of a plant cell to multiply and regenerate into a new plant. What is totipotency? Ability of a cell to multiply and regenerate into a new plant is called totipotency. This is a very important word which is asked in the exam for 1 mark question. What is totipotency? Right? So what is totipotency? It is the ability of a plant cell to multiply and regenerate into a new plant. And this property of totipotency has led the scientists to develop desired plants from explant. Very important word. It is called explant. Here you can see the beautiful picture here, right? You can see the beautiful picture of how they cultivate the plants in the bottles, right? So what is totipotency? Totipotency is the ability of a plant cell to multiply and regenerate into a new plant. As I said, this property of totipotency led the scientists to develop desired plants with explant. As I said, explant. What is this explant then? Explant is part of a plant. Any part of a plant, may it be a cell or a tissue or an organ grown in test tube. So where is it grown? It is grown in test tube. Under what provided with what? Provided with artificial nutrient medium. Artificial nutrient medium under aseptic conditions. What do you mean by aseptic conditions? Sterile. free of microorganisms. Environment which is free of microorganisms, because they may infect the nutrient medium and the whole lot gets destroyed. So what is totipotency? As I said, totipotency is the ability of a plant cell to multiply and regenerate into a new plant, and this property of totipotency has led what the scientists to come up with desired plants from the explant. So what is an explant? Explant is any part of a plant like the cell or a tissue or an organ grown in a test tube provided with artificial nutrient medium under aseptic conditions or sterile conditions. Hope you have understood what is totipotency. This is a very important word from examination point of view. And the nutrient medium is provided with carbon sources like sucrose, inorganic salts, then vitamins, amino acids, and growth regulating hormones, like auxin, gibberellins, and cytokines. So the nutrient medium is provided with the carbon source. As I said, the plant cells are grown in what? Artificial nutrient medium, and this nutrient medium is provided with carbon sources like sucrose, inorganic salts, vitamins, amino acids, and growth regulating hormones like auxin, gibberellins, and cytokines. So the nutrient medium is very, very important for the growth of for the regeneration of a new plant from the explant. Next, the cells divide. So the cells of the explant divide and give rise to undifferentiated mass of tissue called callus. So moving on to what is plant tissue culture? It is a method in which a plant cell or a tissue or an organ is grown in an artificial nutrient medium under sterile conditions provided with controlled conditions of light and temperature. And as I said, what is totipotency? It is the ability of a cell to multiply and regenerate into a new plant. And as I said, the property of totipotency led the scientists to develop the desired plants from explant. So what is explant? As I said, explant is a part of any part of a plant like the cell or the tissue or an organ grown in a test tube with artificial nutrient medium under sterile or aseptic conditions. And as I said, the nutrient medium is provided with carbon sources like sucroses, like inorganic salts, vitamins, amino acids, and growth promoting hormones or growth regulating hormones like auxin, then gibberellin, and cytokines. Moving on to the next, selection of plant matter. Very, very important. As I said, you can see in this beautiful picture how the plants are grown. Right. So the part of the plant used for culture, as I said, is known as explant. Explants are, as I said, they are totipotents. That is the ability, or they have the ability to multiply and regenerate into a new plant. So the explants have the ability to multiply and regenerate into a new plant. So the cells are extracted either from the nodes or leaves, seeds, roots, buds, etc. All these are called as what? Explants. As I said, what is an explant? Explant is any part of a plant, may it be a cell, tissue, or a organ. Organs like nodes, leaves, seeds, roots, buds. And as I said, explants are selected from plants with desirable character. They are very, very important, and they are completely sterilized. Sterilized means they make sure that it is not infected with the microorganism. This is a very, very important condition for a growing of the plant cells in a through an explant, right? So what is very important criteria? selection of the plant material, that is the explant. And as I said, explants are what? Totipotent, right? And as I said, the cells here, the cells of the explant multiply and become and give rise to undifferentiated mass of tissue called callus. Very, very important term in plant tissue culture. Now, I might ask for one mark. What is callus? So, as I said, the cells of the explant multiply and give rise to undifferentiated mass of tissue called callus. And this callus will give rise to embryos, called somatic embryos. They are called as somatic embryos. So, this callus will give rise to what? Embryos called somatic embryos. And this embryos will regenerate into a new plant. So, this embryos will regenerate into a new plant. So, very, very important points to be noted from this very important concept called tissue culture. I repeat, so what is an explant? Explant is nothing but it is a part of the plant. Any part of the plant, may it be a cell or a tissue or an organ grown in a test tube with artificial nutrient medium under sterile conditions or aseptic conditions. And the nutrient medium, as I said, the nutrient medium is provided with what? Is provided with carbon sources like sucrose, then inorganic salts, vitamins, amino acids, growth promoting hormones like the auxin, gibberellins and cytokinins. And as I said, the cells of the explant multiply and give rise to what? Undifferentiated mass of cells tissue called callus. And this callus will give rise to what? Embryos called as somatic embryos. And later the embryos will regenerate into a new plant. And it is this embryos which is going to regenerate into a new plant. So the process of production of thousands of plant through plant tissue is called micropropagation. That is what we are going to study. Preparation of culture medium. As I said, so here you can see the suitable culture medium is essential for the growth of the explant. So as I said, the nutrient medium is very, very important for the growth of the explant. So the explant should be grown in an artificial nutrient medium provided with carbon sources, as I said, like sucroses, then inorganic salts, vitamins, amino acids, auxin, gibberellins and cytokinins. So as I said, medium is prepared using several macro and micronutrients and growth hormones like auxins and cytokinins which promote growth. Agar, I think you might have heard about agar. So agar is added as a solidifying agent. And the culture is autoclaved. So very, very important. As I said, it is done in sterile condition, aseptic conditions. So autoclaving will kill all the microorganisms and it is fully sterilized. So next very important is what? Preparation of culture medium. A suitable culture medium is essential for the growth of explant, and medium is prepared using macro and micronutrients and growth hormones like auxins and cytokinins which promote growth. So agar, as I said, it is one of the very, very important medium. It is one of the important material which is used as for the artificial nutrient medium, as it is a solidifying agent, and the culture is autoclaved, right? As I said, it is autoclaved to kill all the microorganisms. As I said, it is done in what? Aseptic conditions or sterile conditions. So inoculation. So as I said, what is inoculation? It is a process of transfer of explants onto the culture medium. Next, what is done? The explants. It is a process of transfer of explants onto the culture medium which is previously sterilized. So it is done in a special chamber called inoculation chamber, free of microorganisms. As I said, the whole environment where this process is done should be aseptic. It should be in a sterile. It should be sterile. So free of microorganisms. So I think you might have come across many biotechnology labs will adapt all these procedures to come out with the useful plant culture through tissue culture. So next step is what? Inoculation. What is inoculation? The very word it will tell you, introducing. So what is it? It is a process of transfer of explants onto the culture medium which is previously sterilized. It is done in a special chamber called inoculation chamber. And as I said, how is the callus formed? So the cells of the explant will multiply and give rise to what? Undifferentiated mass of tissue called callus. So what is callus then? This is a very important word to be learned. So what is callus? Callus is nothing but an undifferentiated mass of tissue. So it is undifferentiated mass of cells produced by the explants. It is formed within a short period after inoculation. So immediately after the explant is introduced into the culture medium, then the callus is formed. Here you can see how the plants are developed through tissue culture. Then next very important is what? Organogenesis. What do you mean by organogenesis? Process of formation of organs and differentiation. So as you know, cells group into tissues, tissues into organs, and organs into organ system. So it is the process of differentiation of cells into callus, into different tissues and organs. So root initiation occurs. First is very important is what? Formation of root, which is initiated by auxin. As you all know, auxin is a growth promoting hormone, growth regulating hormone, which enhances growth. To cytokinin ratio is very high. So here root initiation occurs if auxin to cytokinin ratio is very high. And shoot, you know, the root always grows below the soil and shoot grows above the soil. For root initiation, auxin should be in higher content than cytokinin, whereas for shoot initiation occurs if cytokinin to auxin ratio is high. Then thus obtained embryonic plants are called as what? Plantlets. So thus obtained, the plants that are obtained are called as what? Plantlets. Next, hardening. You might have come across here, what will happen is immediate exposure of the plantlets to sunlight. As I said, what is tissue culture? It is done in controlled conditions of light and temperature. So immediate exposure of the plantlets. What is plantlets? The obtained embryonic plants through what? Through callus, that is called plantlets. So immediate exposure of plantlets to sunlight, wind, etc. may kill them. So therefore they are grown in greenhouse. You might have come across greenhouses. Many plants are grown in greenhouses. So they are grown in greenhouse for a brief period and then gradually transferred to their natural habitat. You might have come across a word called labland. Gone are those days where we had an idea that we need to sow a seed to get a plant. But now the concept has changed. Science and technology has changed to a certain extent, particularly because of this biotechnology and through tissue culture, we are able to grow a plant in a lab and put it onto the land. So now the days have gone, changed. So where you can see now, those were the days where we had an idea or a concept what we need to sow a seed to get a plant. But now the days have changed where you grow a plant in a lab and put it on the land. So as I said, so immediate exposure of plantlets to sunlight, wind may kill them. So therefore they are grown in greenhouse for a brief period and then gradually transferred to the natural habitat. And this process is called as what? Hardening. So directly we cannot put them on the land because they need to adjust, because they are grown in the lab and they have to adjust to the natural conditions. So that is the reason for some time they are grown in the greenhouses, then they are put onto the land. So this method is called as what? Hardening. So as I said, through this method called plant tissue culture, thousands of plants are produced. That process of production of thousands of plants through tissue culture is called micropropagation. So what is micropropagation? This is a very common word which we use in biotechnology or in tissue culture. So what is micropropagation? The method of production of thousands of plants through tissue culture is called micropropagation. And we find that the plants that are produced through micropropagation are genetically identical. Hence we use the word called soma clones. Or they are also, and why they are called as soma clones? Because they are produced from somatic cells. Because they are produced from somatic cells, hence the name soma clones. So as I said, the method of production of thousands of plants in a short period through tissue culture is called micropropagation, and each of the plants that are produced through micropropagation or through tissue culture are genetically identical, and hence they are referred to as what? Soma clones. And as I said, soma clones, as I said, why they are called as soma clones? Because they are produced from the somatic cells, because they are produced from the somatic cells, hence they are called as soma clones. As I said, sucrose is a source of carbon in tissue culture medium. Hope you have understood what is organogenesis or differentiation. It is a process of differentiation of cells in the callus into different tissues and organs, and root initiation occurs with auxin to cytokinin ratio is high, and shoot initiation occurs if cytokinin to auxin ratio is high. So thus obtained embryonic plants are called as what? Plantlets. And what is hardening? Immediate exposure of the plantlets to sunlight or a high temperature or wind may kill them. So first they are introduced into they are kept for some period in the greenhouses, and then they are brought to the natural habitat. As I said, it takes time for the plants to adjust to the natural environment, as they are artificially grown in the lab, not in a natural condition. We are not sowing a seed to get a plant, and that methodology is called as what? Hardening. And as I said, the method of production of thousands of plants through plant tissue culture is called micropropagation, and the plants that are produced through micropropagation are genetically identical, and hence they are referred to as what? Soma clones. They are referred to as soma clones as they are produced from what? Somatic cells. Hope you have understood the very important concept of this plant tissue culture. Moving on to the application of tissue culture. So transgenic plants, the very word itself, genetically modified plants are called as what? They are called as transgenic plants. What are transgenic plants? Genetically modified plants are called transgenic plants, can be produced by utilizing genetic engineering procedures. Virus-free plants can be obtained. So let us classify them. First one is what? Production of large number of plants from a small tissue or a single cell is called what? Micropropagation. Is called what? Micropropagation. Second is what? Production of genetically identical plants called what? Soma clones. Then third is what? Third application of tissue culture is what? Production of disease resistance varieties through meristem culture, particularly, especially virus-free plants. So that is what is the purpose of producing a plant through tissue culture. So disease-resistant plants, virus-free plants, right? So next, fourth one is what? Production of haploid plants through pollen culture. What is the first one? Production of large number of plants from small tissue or a single cell, where we use the word called micropropagation. All these are what? The applications of tissue culture. First one is what? Production of large number of plants from small tissue or single cell, that is through micropropagation. Production of genetically identical plants, that we use the word called soma clones. Then production of disease resistant varieties through meristem culture, especially virus-free plants. And production of what? Haploid plants through what? Through pollen culture. And next, fifth one is production of somatic hybrids through protoplast culture. What is the fifth one? Production of somatic hybrids through protoplast culture and production of secondary metabolites is very important. So all these are the applications of the tissue culture. What is the first one? Production of large number of plants from small tissue or a single cell, where we use the word called micro propagation. Production of genetically identical plants, that is what we call it as soma clones. Then production of disease resistant varieties through meristem culture, especially virus free plants. Then production of haploid plants through pollen culture. Then production of somatic hybrids through protoplast culture and production of secondary metabolites. All these are the very important application of tissue culture. So as I said, transgenic plants can be produced by utilizing genetic in the manipulation of genes. Virus-free plants can be obtained. Production of haploid plants which are useful in breeding experiments can be produced, as I said. Production of haploid plants through pollen culture. Then production of what? Secondary metabolites, commercially important plants like what? Like we have Taxol, it's a drug, it's a drug. Then we have this vincristine, then vinblastine, then serpentine. All these are the important things, right? The commercially important plant, secondary metabolites. Production of somatic hybrids from unrelated plants which are heat, frost, salt and disease resistant. So it is a technique of multiplying commercially important plants such as orchids. So this is all about the application of tissue culture. This is a very important concept which is asked in the exam. What are the applications of, what is tissue culture and what is the application of tissue culture? Now the types of tissue culture. Now let us study about the types of tissue culture. Here we broadly classify them into three types. One is called meristem culture, one is called anther culture, the other one is called cell culture. So what is meristem? The very word itself will tell you. I think you might have come across the meristematic cells or the meristems, which is usually found in the root tip and the shoot tip, have the capacity to divide at a faster rate. So the rate of cell division is very fast in the meristematic cells, which facilitates what? Growth. So it is the culture of shoot apical meristem, particularly the shoot apical meristem, for the production of large number of virus-free microshoots. So that method of culture of shoot apical meristem for the production of large number of virus-free microshoot is called what? Meristem culture. The very word itself will tell you meristematic cells are involved, particularly the meristems that are found in the shoot apex. As I said, the meristematic cells are usually found in the root tip and the shoot tip. Next, anther culture. So the very word itself will tell you anther is a male reproductive organ. So it is a culture of anthers for the production of what? Haploid plants. And this methodology of culturing the anthers for the production of haploid plants is called anther culture. Then what is cell culture? It is a culture of the cells obtained from callus in bioreactors. That is, bioreactor is nothing but called as what? Fermentation vessels. So that methodology of growing the cells from the callus in bioreactors is called cell culture. So what are the three types of tissue culture? One is called meristem culture, second one is called anther culture, and the third one is called cell culture. So what is meristem culture? It is a culture of shoot apical meristem for the production of large number of virus-free microshoots. What is anther culture? It is a culture of anthers for the production of haploid plants. What is cell culture? It is a culture of cells obtained from callus in bioreactors. Then protoplast culture, as I said, one more. Fourth one is called as what? Protoplast culture. So four types of tissue culture: meristem culture, anther culture, cell culture, and protoplast culture. The cells are treated with lytic enzymes like cellulose and pectinase to digest the cell wall and to obtain protoplast, which can be further grown to form new plants. And these hybrids are called somatic hybrids, and the process is called somatic hybridization. Very, very important culture is protoplast culture. So what is protoplast culture then? It is the cells are treated with lytic enzymes. So the digestion of the cell wall is made by the cellulose and pectinase enzymes to obtain what protoplast, which can be further grown into a new plants. And these hybrids are called as somatic hybrids, and the process is called as what? Somatic hybridization. Then protoplasts are either cultured to get callus or and microshoots or protoplasts of potato fuse. I think you might have come across the plant that is produced through somatic hybridization. Somatic cell hybridization is pomato. Production of pomato through what? Through potato and tomato. Is a classical example for what? Somatic cell hybridization. So what is somatic cell hybridization? Protoplasts are either cultured to get callus and microshoots or protoplasts of potato fused with the protoplasts of tomato to get a somatic hybrid called tomato. So very good example for somatic cell hybridization. All these are the different types of tissue culture. So what are they? What are the different types of tissue culture? One is called meristem culture. What is meristem culture? It is the culture of shoot apical meristem for the production of large number of virus-free microshoots. Second one, anther culture. It is the culture of anthers for the production of haploid plants. Next, cell culture. It is a culture of cells obtained from callus in bioreactors. Protoplast culture. The cells are treated with lytic enzymes like cellulase and pectinase to digest the cell wall and to obtain protoplast, which can be further grown to form new plants, and these hybrids are called as what? Somatic hybrids, and the process is called somatic hybridization. And the protoplasts of two different desired plants are taken and fused to get a plant of a desired variety. For example, like tomato. Tomato is obtained through what? Fusion of the protoplast of a potato and a tomato called as hybrid, that is somatic cell hybridization. It's a very good classical example for somatic cell hybridization. I'll be back in the coming session with a new chapter, chapter 7 called evolution. Till then, goodbye and thank you.